Good afternoon, everyone. I would like to welcome you to today's webinar, Clinical Research from Start to Finish. I'm Jane Dixon Weber, the Director of Education and Support Services at the National Fragile X Foundation, and I have a couple housekeeping items before we get started. These webinars are made possible by your donations. Thank you for your ongoing support. We have about a 45-minute presentation today, and then we'll take questions for 15 minutes or so. Use the question bar on the right side of your screen, and you can type your questions in at any time. Today we have Sharyn Lincoln and Kate Pawlowski of Boston Children's Hospital to speak with us about how clinical trials work, what it takes to participate in them, and what happens after the study. Sharyn received her bachelor's degree in biology and religious studies at the College of William and Mary and her master's in genetic counseling from Brandeis University. She is currently a licensed genetic counselor practicing at Boston Children's Hospital in the Genetics and Fragile X programs. She is also the Boston Children's Hospital site coordinator for the Undiagnosed Diseases Network. In addition to clinical responsibilities, Ms. Lincoln is a member of the hospital's Bringing Expertise to Selecting Tests Committee, the BEST Committee, which reviews utilization of complex laboratory tests. She's actively involved with, I'm not sure how to say that, Sharyn, Brandeis and Boston University Genetic Counseling Programs as a lecturer and thesis committee member. Kate graduated from Wellesley College with a degree in psychology and French cultural studies and is the research manager of the Fragile X program at Boston Children's Hospital. She has extensive experience in coordinating and managing clinical trials in patients who have Fragile X syndrome, Down syndrome, and autism spectrum disorder. Kate enjoys getting to know patients and their families and helping to guide them through the world of research. In her spare time, Kate likes to travel, read, and cook with her roommates. Welcome, Sharyn and Kate.
Thank you. Thank you, Jane. So welcome, everybody. Kate and I are going to go back and forth. So I'm going to start. Just first and foremost, neither of us have anything to disclose. The outline of our talk today is going to start with what is clinical research, then talk about the differences between research studies and clinical trials, what it takes to be involved in research, and then what happens after a study is finished. So as you can see from the word blurb, basically research can include all sorts of things, new drugs, medical treatments, products, testing, trials, and you can hear about placebos and randomized control trials as well. So clinical research is really any research that involves human subjects. So people, no animal models, and there are two different main types of research that involve human subjects. The interventional research, which are like clinical trials, and observational research. There are many federal regulations in place which are all designed to protect the research participants. So again, the different types of research: interventional, clinical trials test a new medicine or device. Therapeutic interventions may be like ABA therapy, sensory-based therapies, and the effect that has on certain populations. Non-therapeutic or observational studies may be where we are actually just kind of watching a population long-term, kind of like the forward database where we're getting a lot of information of what we call longitudinally or over the long run. It can also just involve medical record reviews. Animal studies are those studies in mice and flies and zebrafish. So you often hear about new medications, and they start with work in flies or mice, and then they move on to humans. And then there's the non-human and non-animal research, and this is what we call kind of the basic science research. So this involves molecular analysis, cell biology. Basically the things that we do to learn about the Fragile X pathway are very often in this non-human and non-animal research. So we mentioned that there are a lot of federal regulations in place to protect subjects who participate. So on a local basis, all institutions have what's called an institutional review board, or IRB. And this is literally any institution that does clinical human subjects research. So hospitals, universities, they will have their own IRB. So Boston Children's Hospital has its own IRB, whereas a place like the Mind Institute and Rush also have their own IRBs. The IRB ensures that research regulations and ethical guidelines are followed, and on average, they meet 1 to 2 times a month. The IRB may go back and forth with the study team several times to get to the protocol right. So what often happens is that the study team submits the protocol, basically what is happening in a research study, to the IRB. The IRB reviews it, and they may have changes that need to be made. They say, you know, this is good enough, but we need to make these changes and then we'll approve it. Or it may be that enough things need to be changed that they actually defer the protocol, and a lot of things have to get reworked, and then the study team has to resubmit. The national regulatory groups is primarily the U.S. Food and Drug Administration, or the FDA. And the FDA oversees clinical trials involving new medicines and new medical devices. It regulates clinical trials, and it's responsible for approving or not approving any new medication that comes on the market based on the results of the clinical trials. So any clinical trial has to go not only through the FDA review, but then also has to go through the IRB review. So where did some of these rules and regulations come from that we've mentioned? Well, there are a number of ethical guidelines that have been developed. The Nuremberg Code was developed after the Nuremberg trials after World War II. It outlines that consent or participation must be voluntary. You cannot force somebody to participate in a study. The risk-benefit ratio has to be reasonable. And again, participant freedom. So not only the freedom to decide whether you want to participate in the study, but also you can withdraw at any time. The Declaration of Helsinki also talks about research versus medical care and the risk-benefit ratio. And the Belmont Report is really talking about kind of our basic principles of ethics: the respect for persons, so autonomy, also beneficence, which is the basically, you know, do no harm, you need to provide some good, and justice, making sure that all populations can participate, or that it's reasonable for all populations to participate. So the key to research is the consent, and consent is basically agreement to take part in the research. And again, we can't emphasize this enough, but consent must be voluntary. So there should be no coercion. People shouldn't be paying you exorbitant amounts of money to participate in the study. Your doctor shouldn't be saying, if you don't participate in this study, like, you can't be my patient anymore. And there are special regulations for people who are considered vulnerable populations. So these are individuals with intellectual disability, with pregnant women, children. All of those are considered vulnerable populations. And the consent, in addition to being voluntary, has to be informed, or at least attempted to be informed. So researchers need to explain the study clearly, and families and participants need to have time to ask questions, and should ask questions if they don't understand something in the consent. So these are all the things that are pretty much required to be in a consent form. So explaining what is the purpose or purposes of the research. How long will the study last? It needs to provide a thorough description of the research, as well as any drugs or devices. So this is really, you know, what happens during the research? What are you expected to do, and what are the study doctors going to do? What are any potential risks and benefits? Are there alternatives to the study? And then pretty much every consent form is going to talk about confidentiality and privacy specific to the research study, but also the HIPAA laws in general, who to contact with questions, and there are reminders kind of throughout the consent form that participation is voluntary. We also talk a lot about consent versus assent, and anybody who's participated in a research study with their child may have heard about the assent, or the study providers may have asked them if they felt that their child could provide assent. So consent is given by a legal adult, which is basically 18 years, or emancipated minors, anybody that the court system and legal system considers a legal adult. So parents provide consent for their children. Adults who are deemed competent provide consent for themselves. Assent is given by a child or an adolescent. Generally, about the age of 7 is the youngest we do it, all the way up to 18. And somebody who's not able to give legal consent for other reasons, like cognitive abilities or some other medical reason. And the reason that we do the assent is that it actually does matter. If the person providing assent does not agree to participate in the research, they don't participate in the research. There are some rare exceptions to that, but in general, if you came into one of our studies and the parents were all for it, and we talked to the child or the adult participant and they said, I don't want to participate, we have to respect their wishes.
Awesome. Thank you, Sharyn. That was a fantastic overview of some of the regulations that govern research. So now we're going to talk a little bit more specifically about the types of research that you might be consenting or assenting to. So as we all know, research is a very diverse field, and we're going to talk first about non-therapeutic or non-interventional studies. So what are these? Basically, it's any time that there's no treatment being implemented. So it's not a clinical trial, it's not any sort of therapeutic trial, but that leaves a whole wide range of things open. It can cover topics like all sorts of things from, if you've ever asked yourself, do Fragile X patients have a high incidence of seizures? That would be a question that could be answered by a non-therapeutic study, all the way to something like, how much money does it cost us to treat anxiety? That's another question, very different, but could also be answered by a non-therapeutic study. So there are obviously a wide range of topics, and they can still require quite a bit of effort on the part of families and participants and also researchers. The most common types of non-therapeutic studies are observational studies. Sharyn mentioned the Forward database, which is a joint project between the CDC and the National Foundation, trying to look at every patient with Fragile X syndrome who comes into clinics. That study follows patients over time, and families fill out lengthy questionnaires, but there's no intervention. Researchers aren't doing anything specifically different. They're just following them through their normal care. Another common type is medical history review, review of patients' medical records already in an institution. Other types are quality improvement projects, which you may have seen at your home institution, even doctor's offices. Basically, it asks if we are providing the best care that we can. And then the structure for these studies, there are 3 big types of structures. They can be longitudinal, which basically means over time the data is collected. Cross-sectional, where you look at a lot of people but only at one time. And then a case study, which you may have heard of. They get, if something's very rare, it gets written up as a case study. It's either one individual or maybe just a few people who have a specific condition. So why would you do this kind of study when there are other studies like clinical trials that exist? Well, first of all, it's important that we further our understanding. Basically, everything that we know about different disorders or different conditions has come from trials like this that just take an overview of what's actually going on. They prepare us for implementing clinical trials, and they also improve those trials. So all of the assessments or questionnaires that you've ever filled out before, those all have to be tested as their own research projects. So if you've ever heard of someone taking an IQ test, well, that test was developed and was practiced and was part of its own research study before it ever got used in any other situation. And also because we need to information gather. So there are things that we know and there are things that we think we know, and we need to prove those things that we think we know by doing information gathering studies. And also, like I mentioned with quality improvement projects, one of our goals is always better clinical care. So we can look at standard of care studies, which ask the question of, are we all doing the same things? Then we can look at longitudinal outcome studies and ask ourselves, are those things the right things? So with all of those possible topics, how do you ever actually find a study topic? Well, some study topics come naturally from molecular animal studies like the ones that Sharyn was mentioning. They can also come from past clinical trials, successes and failures. I'm sure a lot of you know about past Fragile X studies that suffered from a lack of good outcome measures, meaning it was hard for us to tell if anything was happening because the tools we were using to measure were not sensitive enough. So, you know, taking that lack of success in a past clinical trial could lead us to say, let's create a better form that captures this information in a way that's more useful. Research in related disorders can often lead us to new research projects. Pilot projects, which is a study of a very small number of people to see if something works, focus groups, and clinician and family ideas. So many different projects have started because a clinician or a family member said, hey, I've got this issue or I've got this problem that's come up more than once. That is the genesis of the next research project. So always keep that in mind. And why would you do this kind of study as a family or as a researcher, especially knowing that there are therapeutic studies that you could be working on? Well, for families, I think a big benefit is furthering the knowledge on the topic because it can help you as a family, can also help other patients. And for some studies that do different research assessments or reports, you may get the benefit of those assessments. So if someone is doing an IQ test as part of a research study, you may be able to receive a report of that at the end of the study. Those reports might have some benefit to you, but it might be mostly that you got extra information on the study, so there can be kind of a balance of different things. You probably won't get any what they call direct benefit the way that you would in a clinical trial. So a clinical trial may say that if you are taking the active medication, it could possibly alleviate some symptoms. In this kind of trial, you don't really, in this kind of study, I should say, you don't really get that benefit. Why are researchers doing these studies? Well, first of all, because we're always hungry for more information. So the more knowledge that we can have on any disorder or any condition, the better off we are. Another thing that's very important is that we always try to do follow-up studies and confirmatory studies, because if you see something one time, although you can be, you do a lot of things to make sure that that result is accurate, you really have to replicate it. Otherwise, it's not worth very much. Studies also always introduce other studies. As you're going along, you say, wow, you know what I realized? Or you know what I wish we had spent more time on? This other avenue. So doing research naturally begets more research. And the last benefit for researchers, of course, is publishing and sharing results. And so that's a nice way that we kind of all benefit. When you are thinking about taking part in any type of research, but especially non-therapeutic research, it's important to understand what the study looks like. So the duration, which is how long the study is going to last. When researchers are thinking about that, they have to balance the long-term scope versus the practicality. So the longer the study stays open and the longer people participate in the study, the more information you get, which is wonderful. On the other hand, it's not practical to have a study that stays open for 25 years and never closes and you never hear any of the results. For design, you're trying to balance efficacy versus, again, practicality. What information is going to be collected? How will it be collected? Is that the most efficient way to collect the information? And then detail. So, once you have a person who's willing to participate in a study, you want to get as much information from them as you can, but you also have to be practical. So how much information is too much information? It may surprise some of you to know that you can actually ask too many questions, and that can be very bad for you in terms of statistics and also in terms of practicality. And you may be asking yourself, where does the time go for these studies that don't involve a new medication or a new interventional treatment? Well, it takes time to find funding and to figure out what's going to be paid for. The Institutional Review Board approval that Sharyn was talking about obviously has very many steps and can take some significant time. Recruitment can be difficult depending on how difficult the study is or how small the parameters for people allowed into the study are. And eligibility. So you may show up and find out that, oh no, unfortunately you can't participate because we have already taken too many boys who are 7 years old. And those kind of fluke things just sort of happen, and that can take a lot of time to kind of get around those things.
Okay, so Kate did a great overview of the non-therapeutic interventions. So I'm going to talk about clinical trials and some of the differences between the two. So clinical trials in a nutshell look super simple. You get an approved protocol, you select your, you get an approved protocol from the pharmaceutical company, you select the people who are going to run the study, you go through IRB approval, you get patient recruitment, the data is entered and reviewed, you have your data analysis, you present and publish your data, and the data is filed, registration for the drug is obtained, and you know, that sounds really simple, looks very simple, nice stepwise manner. It is not anywhere near that simple, unfortunately. All of the steps are simple in and of themselves, but take a lot of time to get through. So developing a new medicine in itself takes a long time. You have the basic science research that needs to be done that even lets you say, hey, a medication that targets this receptor or that targets this particular thing in the cell could actually be helpful. That's been trialed in animals to see if it actually does appear to make any difference whatsoever. Then you get to human trials, and we'll talk about this, but the second point is that human trials have many sub-steps or many phases. So this alone, from the basic science research through the animals, through the human trials, can take many, many years. So you'll hear about different phases for clinical trials. So phase 1 is safety only, and this is often done in a healthy population. Sometimes it's not, but for the most part it's done in quote-unquote normal controls. Phase 2 trials are where we start to get the affected population in. And so the medicine is given to the population that is supposed to help. It is still focused a lot on safety, but it is starting to look at the effectiveness of that drug in the study population and starting to look at potential side effects. If everything goes well in a phase 2 trial, we move on to a phase 3 trial. This is where the drug is given to many, many more people. So in these situations, especially in rare diseases or genetic conditions, a phase 2 may be in an older population, and phase 3 starts to include the younger population. It's looking more and more at efficacy, and it's looking at that long-term effectiveness as well as long-term safety, and they're continuing to monitor side effects. Phase 4, if it goes there, is even larger than phase 3, but it is important to note that not all medicines will require phase 4 trials, and that's certainly the case in most genetic conditions. You're not going to get tens of thousands of people in for most genetic disorders. So this is just a picture of those different phases. So the preclinical is all those lab studies, the animal trials, the things that we talked about. Phase 1, small population, just looking at the safety. Phase 2, you've expanded it, you're looking at the safety, you're looking at bigger populations, and then phase 3 is going to be your long-term efficacy and long-term safety. So making a clinical trial, there are a lot of things to think about in the study design. The first is your hypothesis. What do you think that this medication is going to do for your population? And then the outcome. How are you going to measure what you think the drug is going to do? The dosing is also very important, and this comes into play a lot with the different types of clinical trials. Are you going to get one dose? Are you going to get multiple doses? What happens if you get to a dose that a lot of people are having side effects? Do you come back down to the next dose? All of that needs to be thought of. Also the number of visits, the inclusion and exclusion criteria, and then the study design as a whole, which all of this plays into. And all of this needs to be figured out before a company can talk to clinical sites about participating and before submitting to an IRB. So that's a lot to think about before you even get to your initial trials. So let's say that the pharmaceutical company has figured all of this out. They've gotten their own protocol and everything together. They've approached the clinical sites. So now we're at the point where we're working on getting IRB approval. So we have to draft a protocol summary. We have to write a consent form. Sometimes the pharmaceutical companies do this. Sometimes it needs a lot of work from them. Sometimes it doesn't. You need to find the space to conduct the study. Do you have somewhere in the hospital that you can do the study? Do you have all of the right equipment that you need to do the study? And then you need to make sure you have the right people. For clinical trials, you do need MDs involved. You may need PhDs to do any of the neuropsych testing. You need pharmacy involvement. You're probably going to need research assistants and research coordinators and various other people to help you actually implement this. So you can see why it's already taking a long time to get a clinical trial up and running. And then once we get to the point where the clinical sites are involved, I'm going to go back a slide real quick. We've done all of this. We've gotten the protocol summary consent form. We've got the space to do it. We have all the right people. Then we need to submit to the IRB, and this can take a long time. As I mentioned, it's the schedule of the IRB meetings. IRBs may have changes that need to be made before they give final approval, and there can be substantial differences between the site IRBs. So one hospital's IRB may be much more challenging to get approval from than another one. That may be because they don't have as much experience with clinical trials, or they're dealing with a much more vulnerable population. And so one institution may be able to get approval within 1 or 2 meetings, whereas another institution, it may take 3 or 4 meetings before they get approval. Then once approval is obtained, we still have to go through and get agreements in place between the clinical site and industry sponsor, and that can take a long time because that's all the legalese stuff. Okay, we've done all this, and now the big question is, can we finally start? Well, maybe. So once we have IRB approval, all the appropriate contracts signed, we actually probably still can't start right away. Usually the sponsor will do a site visit to make sure everything is ready to go. All of the people are up to speed on what they need to do. All of them are trained on everything before the site can actually start looking for patients to enroll. So as you can imagine, we may say, great, we've got a great idea for a clinical trial. We talk about it, and a year later, we might finally be at the point where we can start enrolling patients. Now we get to the trial in action. So we have the screening versus enrollment. So as Kate was mentioning with regards to kind of eligibility and everything, that often takes place in the screening visit. So we may think that, yep, you sound like you would meet criteria for this study. And we bring you in for a screening visit where we go through all the inclusion and exclusion criteria, and you've gone through the consent, and we're going through certain evaluations, and we find out that you aren't actually eligible for the trial. So that can very well happen. If you are eligible, then we enroll you in the trial. Participants can withdraw at any time, and then more people must be enrolled oftentimes to finish the study. After the first participant completes the study, it could still actually be quite a long time until the entire study is finished. So let's say that person A starts a trial today, but we don't enroll person Z in the trial until a year and a half from now. Well, the trial isn't going to be finished until person Z finishes all of their evaluations. So you may be waiting another year and a half or so after you've finished the trial before the trial is actually done. Sponsors are not required to provide the drug after the study finishes, even if there is proven benefit. That's a really big thing, and it's very hard for clinicians and for families who notice a difference on the study drug, because the sponsor can say, we're not extending this. We need to do more trials, and we can't give you the drug in the meantime. this is also a really big category, the successful versus failed trial. And a lot of people have heard about the failed trials for some of the drugs that in Fragile X. Now, this successful versus failed is actually, it's objective but kind of subjective at the same time. It's based entirely on the hypothesis, which then is dependent entirely on the outcome measures. So if you choose the wrong outcome measures, you can have a failed trial, but it doesn't mean that the medicine didn't work. It just means that the hypothesis wasn't proven. So failed trials can give helpful information for future studies to design different hypotheses and use different outcome measures. So on the flip side, having a successful trial doesn't mean that further trials will happen. That can be for a number of different reasons. Either the pharmaceutical company decides that they don't want to put more effort into this particular medication because maybe it's not going to reach as large a population as they hoped. Maybe there were some other issues that came up in the meantime with production of the drug or access to the drug or something that is preventing us from doing additional trials. So just because it's failed doesn't mean it doesn't work. And just because it's successful doesn't mean that it will continue in the trial and ultimately get approved.
Awesome. So now if Sharyn and I haven't discouraged you enough on the timeline and you've still decided that you'd like to be involved in research, let's talk about practically what it takes from a family perspective to be involved in clinical research. So you've decided you wanted to participate in a study. So the first things I would consider are, first of all, how do you even find a study? How do I find that there are trials going on that you might or might not be eligible for? What do families actually have to do in this particular study? And as a family or as an individual, can I do those things? You know, can I make myself available for those activities? So first of all, how do we find a study? Well, there are a lot of good ways to kind of keep up with the pulse of research and to hear more about what's going on. One of them, of course, is clinicaltrials.gov. Every clinical trial has to register on clinicaltrials.gov. So if there is a medication trial that's coming up, it has to be on the website. It is up-to-date information. When you go to the website, you can search for Fragile X syndrome. You can search for other terms. You can look by the region that you're in if you're not willing to travel or if that's not something that's possible for you. I would say don't forget to look up related disorders. So if you're looking for your child who has Fragile X syndrome and they also have a diagnosis of autism, I would see what's going on in the world of autism. That can be true for, kids, if you have a child who, oh, also now that I'm thinking about it, they have ADHD. Maybe there's an ADHD trial. Those related disorders can give you a good sense of what's out there and maybe even what's kind of coming next in specific conditions. I would suggest also follow and get involved with local or national groups that will kind of keep tabs on research for you. FRAXA is a research group. The Fragile X, National Fragile X Foundation's CSN groups, your local family support groups, they also are totally up to date on what's going on in different fields of research. Social media is actually a great way to keep connected. A lot of clinics and clinical trials will advertise on social media. There may be discussions on partial results and things like that, so that's also a good way to keep in touch. Your clinic contacts, if you are near to a Fragile X clinic, keep in touch with them, see what your home clinics are up to. You can check out the FXCRC, which is the consortium of Fragile X clinics across the country, and they will also have some information on how to be in touch with your local clinic. And research registries. This is my personal favorite because you can make the projects come to you. If you take part in research registries like the Forward project that we've talked about, those researchers who have a project that you might be eligible for, you're the right age, you have the right diagnosis, things like that, they will actually contact you about research studies, and if you don't want to do them, then that's fine. And if you are interested, then you can get more information. So personally, for me, as a person who's a little bit lazy, I love research registries. Just here are some examples of some of the research projects that we are doing right now here at Boston Children's Hospital. In Fragile X X syndrome specifically. We have a study that's looking at sensory processing that is all online surveys. We are also looking into what we can learn from miRNA, and we also have a new clinical trial coming soon. That's a little teaser. We have some autism-specific studies as well, and if you have questions about any of the research that we are doing, my contact information is at the end, and if you have any questions or comments about it, I'd love to hear from you. Now, let's say you found a study and you're trying to look into it a little bit more. The study should describe the visits themselves. What are you going to be doing during the visit? Are there tests? Are there assessments? Are there other activities? What is the timing of the visit and the scheduling? Is there payment or reimbursement? Is there compensation? So what's the difference between those? Payment and reimbursement. Payment is anything that you're given as kind of a gift for the study. Reimbursement is anything, obviously, that you spend that you need to be reimbursed. And compensation is compensation for your time. So if you have to miss a whole day of work, are you going to be compensated in any way for that? And just a reminder that even clinical trials are not only in clinic. They can also involve home components like phone calls, online forms, questionnaires, and even in some trials, at-home study visits. Okay, so I found a study that I really want to do. Now, what should I do? So first of all, get involved and get connected with the study team. Here are some questions that I would ask anyone, and I love when I hear these questions. So, what are the time commitments? Be specific, because it may say that we're going to be doing IQ testing and this other blood work and this and that, but I don't have an idea of what actual time that's going to take up. Well, the study team will be able to tell you. What are the exclusion criteria? What are the chances that, as Sharyn talked about, I go in for my screening visit, but then I'm unfortunately not eligible to participate in the main trial? What are the next steps from this study, and when are the results going to be coming out? And then the other thing is just preparing for success. You know yourself and you know your family members better than any of us, and so know what you're getting into, and practice makes perfect. If there's something that you are not super comfortable with or super familiar with, the more exposure you get, the better. And then what should you tell the study team after you're done asking all of your very informed questions? Well, you are the expert when it comes to your child. If you are confused or uncertain about anything, if you have any specific concerns or questions, ask. That is why we are here. This is what we're doing. We are committed to making sure that research goes as well as possible. In that vein, we had a family come in for a clinical trial here at Boston Children's, and when they first came in, there were some concerns that they might, that the adolescent who was participating in the study, he was a little concerned, his parents were concerned that he might not be able to complete all of the activities. He was very nervous about some of them. The research visits themselves were very long days, and we were all worried, will he be able to get through everything? And, wouldn't you know it, he was a total champ, and he overcame a bunch of fears that he had during the course of the trial. He got much more comfortable with coming into the hospital, and it was definitely a really rewarding experience for all of us. And it was a great reminder that, with patience and practice, you can get through different things, and even if you are not totally certain, there's no reason kind of not to take the leap and to try to participate in things. So just kind of an optimistic success story.
All right, so now we've gone through all of the pre-steps, and we've gone through the IRB, and we've done the research study, and we've done the trial. So what happens after all of that is done? So the big next step is data analysis and then hopefully publication. So data is generally analyzed after the last participant has fully completed the study. Most of the time we do not go through data analysis prior to that because we're going to get skewed data, and that data isn't going to be published or anything. We need the full data set. There's also a data cleanup and review process where we actually go through and make sure that the data is accurate. The cleanup process can sometimes happen during the study where you're looking through and you're saying, oh, well, maybe we're not all answering this particular question the same way. But that also happens at the end just to make sure that the data you're analyzing is the best that you can do. And you can analyze the data in many, many different ways. So you can have statistical analysis, and you can have what's called qualitative analysis, where you're looking at themes. And a lot of times, the data may be analyzed in a way that makes it look really good, even if it's only okay. Before publishing, if we do publish, we have to write the paper, you get feedback from the authors, you submit to a journal, you get feedback from the journal and the submitting edits. So even if a paper is accepted for publication, it takes a while for that process to happen, and then it doesn't even appear in the journal immediately. So once an article is accepted for publication, it usually shows up online within the next month, but it may not show up in paper copy for a couple of months. So when do we see the results, and we meaning the participants? Well, the information that gets returned to families depends on the study. Not every study is going to return every research result to you. It could be that you may not get any of the results back until everybody else gets the results in terms of a publication. But if you're not hearing anything and you're not sure if you get any results back or not, you can always contact the study to see if there is any new information. And you can do that regardless. So if you have a question as to how the study is progressing, when is it going to be done, how is the data analysis going type of thing, you can always contact the study. Some studies make the news. We know that sometimes they get into the New York Times or the Boston Globe, and sometimes they actually make it onto national TV, more often local TV, but sometimes the big ones get onto national TV, and then we get super excited when that happens. So after all that happens, is that really it? Well, as Kate mentioned earlier, research always leads to more research. We almost always come up with new questions as we're doing a research study. Basic science research may lead to an observational study or to development of a new drug. Trials may go to the next phase of clinical trial, and observational studies may lead to further projects. And I think what's really important in what we've seen recently is that a failed trial may lead to a different trial. So summary and conclusion is that research takes time. If nothing else, research takes time. There are many different types of research that take place, and the non-human subject research is just as important and supports the human subject research. Participation may not benefit you directly, but it can benefit the Fragile X population as a whole, or lead to studies that might provide you with direct benefit. So at this point, we do want to open it up for questions, but anytime that you want more information about clinical trials, you can go to the sites that Kate mentioned. We've also just published an article in the Foundation's newsletter, so you can see a lot of the information that we talked about, as well as some more details on some of the things we talked about in the article. And then you can always contact us with any general questions about research. If you have questions about our clinic, you can contact me. And if you have questions about clinics that are close to you, if you're not in the Boston area or New England area, you can find that information on the Foundation's website, and Jane is a great resource for that as well.
I would thank you for this. This was very informative. But the couple things that I want to add is that the Foundation now has a new website, and we do post every clinical trial and study that we're aware of or that we've been asked to advertise, and that's on our website under research. That's under research recruitment, under research.
And very often that information will also go out to the listserv as well in the email.
Yes, it does. It does. And we post it in our newsletter. I do also want to let families know that anytime someone asks us to post any research recruitment, if it involves like filling out a survey or even a phone call, I do review all of those before they're sent out to or posted on social media or posted on our website. So I know it's already gone through your IRB, but in many cases it has to go through Jane too. I just want to make sure. It's again, some I want to make sure that they're sensitive to families when they go through them. So that's worked out pretty well. So most of them that's posted, I've gone through them all, or I've talked to the professionals who's ever done that. So I've got another question for you. If I'm not sure that my child can participate, what should I do?
That's a great question. I think that's a question that is hard for us to answer in a lot of ways because, of course, everybody is so individual. I think in some cases we kind of luck out in that people are asked to do similar activities in studies that they're already asked to do in their, in their regular life. So you might know that my child is, terrible at giving blood draws, but really, really likes to do psychometric testing, loves going in and playing with the toys and being with the psychologist. So when you hear about a study that has no blood draws but has psych testing, you already know, oh, I think, my child will probably be very good at that. On the flip side, you may be hearing about studies with activities that you never come across in your normal life. And for those studies, I think part of it is talking to the study staff about the supports and the training that they can give you and that you can pass on to your child. And just keeping in mind that, researchers, we spend a lot of time talking about Fragile X, meeting with families. We do understand the concern. So if there is a situation where you get somewhere and you're psyched to participate and it's just not a good day or something goes wrong, these are all things that we are anticipating already. So really, there is no harm in trying something and having it not work out. And we are here to help you and we're here to support you. If you do decide, I want to try this even though I have no idea how it's going to go. So those are just a couple of things. Sharyn, do you have anything?
No, I mean, I completely agree. I think reaching out to the study staff and saying, how can we make this easier? I know that some studies have put together like social stories about being in the MRI machine and that sort of thing. So they may have already thought through a lot of the big challenges. So definitely reach out to the study folks. We're pretty flexible, and certainly if it takes place at a site that has a Fragile X clinic, we're super familiar with the issues that come up. And we see it in clinic. I mean, an example is, this wasn't a research study, but we had a family coming to clinic a number of years ago, and they were worried that their child wouldn't even get out of the car. And we said, that's fine, we'll come downstairs if that happens. We'll come and see you guys at the car. So, it's always worth trying.
Mm-hmm. We're ready for the eventuality, so we can work together. We really do, at the study team, we always think of ourselves as in a team with the parents and with the family. So
very nice. Let's see. Kate, can you explain the second research piece that you had listed? I think it was mRNA. What is the test?
Actually, I knew that was going to come up.
I'm gonna punt that one right to Sharyn.
So I don't know tons about the study itself, but miRNA stands for microRNA. So kind of as a little bit of background, you hear a lot about DNA and the genes, and you hear about the protein. So for Fragile X, you have the Fragile X gene, FMR1, and you have your Fragile X protein, which is FMRP. In between DNA and protein is a step called RNA. And it's basically very similar to DNA, but it allows the protein-making machines in the cell to actually be able to make the protein. So it's basically kind of how I explain it. You transcribe it and then you translate it. So it's like taking American English, transcripting it or transcribing it to British English and translating it into French. So American English and British English are pretty similar, but there are some words that are really going to throw you for a loop if you don't know what they are. So it's kind of the same idea. There are different types of RNA, and microRNA is one of them. And it is of particular interest in the scientific community because it's thought that some of those RNAs can kind of inhibit things and basically affect the way other proteins work. And so that's one of the things that a lot of researchers are looking at, not just for Fragile X, but for other genetic disorders as well, as to whether the microRNAs may actually be able to either inhibit overproduction of proteins or potentially in some ways kind of help ramp up the activity of proteins that need it. So I couldn't tell you what our study is looking at specifically because I haven't been involved with it, but that's kind of what microRNA is at this point.
That's a great summary. I always go to Sharyn for all of my biological questions, and I will say, of course, for that study or any other study, if you shoot me an email, I'll get you probably more information than you could ever want.
That's pretty funny. Another question that came in is, how do you know if research is safe?
That's a really good question. So I think that's something that's very important to think about. It's certainly something that we spend a lot of our time thinking about. Sharyn gave a rundown of some of the ways that we try to make sure research is as safe as possible through those human subjects protections, going through multiple layers of having multiple organizations review the research. So those are some safeguards. If you're in something like a clinical trial, they do interim analysis that goes to something called a data safety monitoring board, which is specifically an outside group that's just watching to make sure that there are not too many adverse events or side effects to the medication. So those are some of the ways that we try to make things as safe as possible. Also, like Sharyn talked about, all of the earlier phases of the trial, the preclinical phases in animals, phase 1 trials, which are only looking at safety, phase 2, which still has a very heavy safety bent. Some of the testing that we do during the course of a clinical trial is not for the outcome and to look at if the medication works, it's only to make sure it's still safe. So you might have a test that looks at your heart activity. You might have different blood tests that are only looking to make sure that you are still staying safe and healthy.
Right. There are also regulations in terms of how much blood can be drawn at any given time and over a period of time for different populations in terms of weight. It's primarily weight-based. If there's radiation involved, like if there are X-rays or if there are PET scans, CT scans, the study has to be reviewed by the Radiation Safety Committee to make sure that it's not a ridiculously large amount of radiation with no, that again, kind of the risk is outweighing the benefit. And the IRB takes into account all of that sort of stuff as well when we're reviewing studies. We take into account the inclusion and exclusion criteria. We make sure that if there are studies that need certain exclusion criteria, that they get put in. So it all falls under those regulations and then some guidelines within each institution as well.
So
and the consent form, before, and on top of that, all of that information is in the consent form. They tell you what the potential risks are in terms of the blood draw, in terms of any imaging testing or any other testing that's being done. So you can determine whether those risks seem to be too much or whether you're okay with them.
So can I assume that the IRB has looked at all that when they went before they gave their approval?
Yes.
So if you have IRB approval, they've really taken all the safety considerations for
they have tried to, yes. I think there's always the possibility of an unforeseen event, an unexpected event, which can certainly happen in any research study and in clinical trials. When you have a new drug, you may not know all of the potential side effects, but that's in the consent form also. And yes, the IRB takes into account all of this.
So the other thing that It brought up the question when you were talking about the design of clinical trials, how you have to state what you think the outcome will be for the trial. And if we, like, we'll use Seaside as an example, they said, this is what we expect to see when people take this drug, and they didn't see that. So what's the chances, or is there any work at the FDA level to say, to leave that more open-ended, so that you might say, well, we might see changes in behavior, or we might see reduction in anxiety, or we might see more social skills, so that it's not just, let's say, for example, it's not just social skills we might see, but what if we see some improvement in behavior? I mean, is the FDA flexible on that at all? At this
yes
point?
And no. It depends on what the hypothesis is. And you can have more than one hypothesis. So if your primary hypothesis is that you're going to see an improvement in social skills, and your second hypothesis is that you're going to see a decrease in anxiety, and your third hypothesis is that you may see an improvement in behaviors, then yes, the FDA takes into account all of those sorts of things depending on if you actually saw changes in any of that. But if you only have one hypothesis dealing with the outcome and you don't reach that outcome, that's all they care about, is that your hypothesis was not proven. Now, you can go back with the same drug and do another trial and say, we saw improvements in anxiety and behavior, so now we're going to focus on that as our hypothesis. But that's also where the outcome measures come in as well. Part of the issue with the Seaside trial and some of the other trials is that we did not have the best outcome measures. We know our kids with Fragile X don't test well. And so if you have a super test-heavy study, chances are you're not going to see a great improvement because our kids don't test well. So you need to make sure that you're using the right outcome measures to prove your hypothesis. And that's something that the FXCRC has been working on a lot, and we now have the Clinical Trials Committee who's working with pharmaceutical companies to try and make sure that they are using better outcome measures.
Yes, so so I am on that committee, the Clinical Trials Committee, and so we are working, and so the attendees know that we are, where in the past, I mean, that's a really good point, Sharyn, but the pharmaceutical companies would develop these clinical trials, not completely, but kind of in isolation. They would say, oh, we think this medication is going to affect with social skills, and here's our trial, and so they get it approved. But now the pharmaceutical companies are working with this Clinical Trials Committee, who's made up of many of the doctors, the Fragile X doctors across the country, and so the Fragile X doctors are helping the pharmaceutical companies set up their clinical trials to try to avoid the Seaside kind of thing. So going back to did you get the IRB approval before you get the FDA approval, or are they simultaneous?
No, you usually need to get the FDA approval first because you need to get what's called an IND, which has to deal with investigational drug status. So you can submit that to the FDA and then work on your IRB process at the same time, but the IRB will not approve the study until they get the IND.
And we should clarify, too, that there are many steps of the FDA approval. So the final approval in terms of actually getting a medication that you could buy at a local pharmacy comes way after the IRB approval and the whole clinical trial. But the very first step, the kind of the FDA saying we're aware of these trials that they're going to be starting, has to happen before the institutions can review the protocol.
Yep.
So do you guys actually go before, like, is it an in-person meeting or is it all done over email?
No, it's so I'm on an IRB. I'm not on the children's IRB. I'm on a different IRB. It is an in-person meeting, but it is generally a closed meeting. So you do not have the investigators there. They do not know what really happens behind closed doors, other than the fact that they get kind of a letter back from the IRB outlining, questions or concerns that the committee had. Very rarely the IRB may ask a researcher to come to the meeting if it's a particularly tricky study, and that it would be helpful to have them there, but that doesn't happen often.
So that's at the when the IRB approval process.
Mm-hmm. Correct. So basically what happens is that the research team submits their protocol summary, a detailed protocol, and their consent form, anything that's going to be involved in the research study. So any questionnaire, any flyers, any general emails, like recruitment emails, all of that needs to go through the IRB. So that gets submitted to the IRB, which then looks at it in their office, determines whether it needs to go to full committee, or whether it can go through a process called expedited review. Certain research, like medical records review, often can go through expedited review, but human subjects research, where that involves people, usually goes through the full committee. The full committee meets. There's a primary reviewer and a secondary reviewer for each study. They're the ones who review the protocol in detail. All of the committee members can read through it and everything, but you have the two people who are reading through it in great detail. They are paying attention to everything. And then it is discussed at the committee meeting. They go back to the IRB goes back to the office, writes up their letter to the researchers, who then need to generally reply back to the IRB with any of the modifications requested. And once those modifications are in place, if they're all okay, then then it's approved.
So that's, I mean, I can't believe how complicated that is. I mean, and that's, all we hear about are IRB approvals, not the well, and so is that what it's like at the FDA level too?
Or I believe for the review of the drug, yes. So the FDA does kind of does multiple different reviews. They do like the IRB review before a trial even takes place. And then they do the reviews after the trial takes place as well, in terms of reviewing the data and everything. And certainly if they're trying to push to market, the FDA has to review all of the data from all of the trials that have gone on before they give final approval.
But
for it to go to market.
But so when you're still developing the trial, so you go to the FDA, I guess in person or emails, and get your IND, is that what you said? And then you take that to get your final approval from your IRB?
Yep.
And then you do the trial, and then the information goes to the IRB and then back to the FDA?
No, the IRB does not necessarily need to review anything once the trial is finished.
Okay. So that all the information, once the results have been in and it's been analyzed, and you want to take it to the next step, if you want to take the medication, let's say public, then that would have to go before the FDA?
Correct.
Okay. Wow.
This is why it takes a really, really long time to get any drugs through. There have been a few exceptions with getting drugs to market relatively quickly, and I would say that the FDA is starting to be a little bit better with orphan diseases, which are basically rare diseases, and nearly every genetic condition is considered a rare disease. Fragile X is considered an orphan disease just based on the population numbers. Yes. So they are getting better in terms of approving those drugs faster, but you still have to go through the steps.
So what about an existing drug like either metformin or the tetracycline products that they find may have benefit in people with Fragile X syndrome? What kind of since the drug is already there, does that just mean you still start you skip the mouse models and you just start with the well, just tell me, so where would that start if you have an existing drug?
Yeah, so you would still want to at least have the data showing that, something like metformin, why it might potentially be beneficial in Fragile X. So you still at least want that background data. You still need to get the okay from the FDA because you're using it off-label.
Right.
And you still have to go through the IRB and everything. But in terms of getting approval at the end of the day for use in Fragile X, that would likely be a much shorter process.
Well,
because it's already on market, people can technically use it off-label now.
If their doctor will prescribe it for them.
Correct.
Okay.
The other thing that we should mention is that you would still have to do the safety data if the dose was going to be drastically different than what you would take for other uses or something like that. So there are cases. So for example, you might be able to skip a phase 1 trial if the doses will be similar to, let's take minocycline. If the dose is very similar to what's being used for antibiotics versus treatment in Fragile X, then you might be able to skip over the safety data because it's already been done.
Right.
But if you find out, no, actually you need 3 times the dose than that's usually prescribed for antibiotic purposes, you would have to go all the way back and start the safety data over again, just because it would be totally different than presumably what was looked at before.
That makes sense. What's the process for finding I'll say, Fragile X uses for existing medications? Like, how would they find that metformin might work or that the tetracycline, like minocycline, might work? How do they, how do doctors find that out?
That's a great question. So there are, I'm not familiar with them, but there are some things that allow you to basically test multiple compounds in like a mouse model, a fly model, or on like a nerve cell, kind of the basic research to see what the effects are. So that it kind of is a crossover between like your basic science research and your clinical trials. So we're probably not the best people to answer that. I'm sure there's somebody who is, but it's not either of us.
We should also say sometimes these things are kind of also found out by accident.
Yep.
So you take a medication for one thing and you realize, oh wow, this medication totally cleared out my acne. And then all of a sudden the researcher says, wait, that's a great idea. And they start a trial that way. So sometimes that's possible, but there are also, like Sharyn said, people are doing preclinical studies looking at how something is affecting a cell, and they might say, oh wow, it's affecting this part as well, or they can say, based on the shape of the molecule that we're talking about, we would expect that it might also do this too. Very, very, very smart biochemists working on these projects is basically where I'm going to come down on that.
And you can also, you can search formularies, which are the giant books of medications, basically, looking for things like, searching for keywords, ABA agonist, or GABA agonist, AMPA agonist, mGlu-R receptor. So you can kind of, because all of that background data, all of the chemical data and the biochemical data and that sort of thing is somewhere. So you can search for it kind of that way too, if you have a lot of time.
I'm with you. That's Well, this has been very informative. I don't have any other questions that have come in, and but I really liked hearing about all these different. I mean, I think it just gives all of us a greater understanding of really what goes into creating a new drug and why it takes so long. And I mean, but there, you know, people are looking out for our safety. You know, it's so it's. You kind of never know what that's going to be. So with that, I think that we can close down for today. I wanted to again say thank you for sharing your expertise with us. I found it incredibly informative, and thank you. And I do want everyone to know that the article has gone out that accompanies this webinar, so it will have additional information. You've got their contact information if you have questions. You know, and along this note about the drugs, every year, and we do it in October, we have Dr. Craig Erickson come in and talk about medications, and so we can ask him more about off-label use of existing medications to see how they come up with that.
Yes, absolutely.
So, and I want everyone to know too, we are scheduling a webinar on Fragile X syndrome and autism. So watch your emails for upcoming webinars. We do, they are made possible by your donation, so go to fragilex.org and make a donation. And so thank you, Kate and Sharyn. Do you have any final closing words?
No, thank you guys for listening, and we hope that we've inspired you to, you know, give research a try if you never have before.
Or at least ask a lot of questions about it.
There you go. Thank you, ladies. Thank you, everyone. Have a great day.
Thank you.
All right.
Thanks.